Unfortunately, as it is, humans contaminate the environment and destroy ecosystems through numerous ways of pollution from agriculture and industry. A common area of accumulated pollution is in water. An example of this, with very obvious and detrimental effects, is oil spills. Bioremediation is an application of biotechnology involved with toxic waste removal in the environment and is one possible method of oil spill cleanup. Bioremediation specifically uses biological and microbial methods in order to convert pollutants into harmless products found naturally, like CO2 or other nutrients. Typically in oil spills, this involves using microorganisms in degrading harmful hydrocarbons compounds originating from petroleum, also known as crude oil. Petroleum is the oil in oil spill compounds, and is most well known for being the material that is used in the production of fuels like gasoline and diesel. Since the oil industry is so important to human society, the transportation of petroleum is not only inevitable, but extremely frequent, with a risk of an ecosystem-changing spillage each time. As such, bioremediation is an area that is constantly looking to be improved. In today's video, I'll give a brief overview of some of the more intricate details about bioremediation. While there are many types of bioremediation using different organisms, I'll be focusing on bioremediation using bacteria specifically, and on the genus Bacillus. Species from the genus Bacillus are shaped like rods, are gram-positive, meaning they have a thick layer of peptidoglycan, and are one of the few types of bacteria that can form endospores. While some are pathogenic and can cause disease in humans, like Bacillus anthracnose and food poisoning and Bacillus cereus, other species are often used in the industry or for agricultural medicine and in biotechnology. Bacillus species are known to be able to degrade organic compounds in oil. They have tolerance to external conditions, have fast reproductions in cultivation, have the ability to sporulate, and their cheap production makes them excellent candidates for bioremediation. Bacillus subtilis is not only one of the most well-studied species within the genus Bacillus, but it is also a modern organism across the studies of bacteria. They are found widely in soil, aquatic environments, and in the digestive tracts of animals like humans and ruminants, despite not being categorized as a pathogen. In fact, if you've ever had an introductory microbiology lab, chances are B. subtilis is one of the microbes that you've seen and worked with. As in line with other species in Bacillus, they are tough as a result of their abilities to survive their environmental conditions. While in the past they were mostly considered an obligate aerobe, meaning that they required oxygen to grow, more recently there has been investigations that study the anaerobic growth of B. subtilis, indicating that they're actually a facultative anaerobe. B. subtilis are also halotolerant, meaning that they are able to survive in tolerant environments with high salinity using mechanisms that adapt to osmotic stressors. All in all, Bacillus subtilis are very useful in both industrial applications like enzyme production and plant growth promotion, and in research like in genome, biofilm, and endospore investigations. So let's talk a little bit about how Bacillus subtilis is able to degrade oil pollution in bioremediation. Within the chemical composition of petroleum/crude oil, there are many different types of hydrocarbons that occur together in mixture. This includes compounds like single-bonded alkanes, cycloalkanes, as well as cyclic carbon compounds called aromatics. It's good to remember when using bacteria in application, it's their biological processes and metabolic adaptations in nature that are being exploited. In studies done about biodegradation with bacterium, the target bacteria for investigation is often extracted from areas of oil pollution. And these bacteria have shown better hydrocarbon degradation compared to those from cultured colonies. Bacillus subtilis and other oil-degrading bacteria are able to break down and use components of petroleum as both an inorganic supply of carbon as well as an energy source, showing a case of chemoautotrophy. Substances like biosurfactants, fatty acids, etc. produced by Bacillus subtilis are able to solubilize oil alkanes and improve many other factors in bioremediation. Biosurfactants typically are important and are linked to increases in hydrocarbon degradation in Bacillus subtilis. This is because biosurfactants are able to increase the total surface area of the contact between water and the hydrocarbons, also called the interfacial surface area, and allow the bacteria to grow on the hydrocarbons. Increasing the space for Bacillus subtilis populations to spread and grow, and the more direct contact of the bacterium and the oil are some of the properties of biosurfactants that allow for more hydrocarbon degradation. However, the major contributing factor of biosurfactants in hydrocarbon degradation is the fact that it is a surfactant. But what are surfactants? Surfactants are compounds that are able to decrease the surface tension between two liquids and can improve the mixing of liquids that are usually unmixable, like oil and water, by stabilizing emulsions of the two particles. Biosurfactants are microbially produced compounds that are more ecological and environmental friendly compared to chemical surfactants, and as such are more favorable to use. Bacillus subtilis can make many types of lipopeptide biosurfactants, and surfactant is considered one of its most effective ones. Two other attributes of Bacillus subtilis that show promise in benefiting bioremediation are their ability to live in biofilms and their formation of endospores. Biofilms are communities of bacteria that live within a sticky matrix of self-made molecules called polymers that let them attach to surfaces. Biofilms act as layers of protection for bacteria by resisting stressful environmental factors and have shown the ability to degrade toxic pollutants. Endospores are a type of dormant and extremely resistant cell state that Bacillus subtilis can go in during stressful environmental conditions. Endospores allow Bacillus subtilis to survive in toxic environments and without extra nutrients. The endospore can then germinate when conditions are favorable again, which also acts as a screening method of the environment's toxicity. Biofilms and endospores are useful because of their living qualities that let them self-regenerate, self-regulate, and respond to their environment, allowing for the survivability of bacteria populations on a site of cleanup. I hope you enjoyed this introduction of bioremediation using Bacillus subtilis, and thank you for watching.